Okay, thank you very much, Chris, for the kind introductions. Okay, good morning, everyone. So we are on our last leg or in our final installment of this webinar series on the metabolomics approach and its different applications. So I hope um, you had been able to learn a lot from the first two sessions, and hopefully in this last session you will also get to do so. Okay, so in the past sessions, so for in the first session, we have covered the introduction to metabolomics and its applications. So we have answered the what, why, when, and where of metabolomics. We have also covered the hows of metabolomics in the second session last 13th of March. And from there, you were able to know some parts of the metabolomics workflow up to the data acquisition. And so for today, we will cover more on the chemometrics in metabolomics and beyond. So for this particular session, we'll cover more of like the data analysis as well as the what are the subsequent steps of the metabolomics workflow. So again, our title is Chemometrics in Metabolomics and Beyond. So as mentioned, our coverage will be chemometrics plus the rest of the metabolomics workflow. Okay, so we have covered more of the definition of the problem, the experimental design, sample preparation, and data acquisition in the previous session. Today, we'll cover more of the second half from the data processing up to the validation part. So, from the data acquisition, we go to data processing. So in we know, right, in chemical analysis, sample preparation is very important. Um, uh, it is known that a lot of the analytical variation comes from the part of the sample preparation, and so it is very important that we optimize the sample preparation in order to get accurate and meaningful data. The same goes for data analysis or or the chemometric analysis. We need to do um, uh, proper and appropriate data pre-processing before we subject it to subsequent data analysis, or in this case, in our case, we will be using chemometric analysis. And here, this is where like peak alignment, things like peak alignment, data binning, baseline correction, noise removal, smoothing. If you have some fluctuations in your signals, you have to make use of like smoothing algorithms to smooth that out. You can also have your variable selection to ensure that you get your the variables that are related to your samples rather than your background or contaminant signals. You also can do like some initial outlier detection, but you can also have this outlier detection part during the chemometric analysis part as well. But if you already know that there are some samples that are outright can be rejected in the pre-processing step, then you can also do so. Okay, so we'll talk about data pre-processing. So NMR-based metabolomics and MS-based metabolomics will have different pre-processing steps. And so now we'll cover first more of the NMR data pre-processing. So, usually there will be like some chemical shift variations in your NMR spectra, and this can be due to a number of things. It can be due to changes in pH, it can be due to differences in concentration, it can be because of protein interactions. And usually, you have to perform an initial chemical shift calibration. And this is done using an internal standard. So, for example, your internal standard will have a peak at the 0 ppm, so you can adjust the chemical shift of that particular compound to the 0 ppm, and that will help align or help calibrate the rest of your spectra. You can also make use of known signals. So there are compounds that we know has a specific peak at a particular chemical shift, and so you can use that for the calibration. For example, glucose has a peak at 5.23 ppm, so you can calibrate your spectrum at that particular chemical shift. Um, you also um, so these internal standards that is commonly used include your tetramethylsilane for example for deuterated chloroform. You also have trimethylsilylpropionic acid and DSS for deuterium oxide. You also have this one is a bit more new, but its advantage over TSP and DSS it has less interactions with proteins because TSP and DSS can have some interactions with proteins. They can also have a bit of chemical shift variations because of pH, but more so for TSP. Um, so this DSA is actually another internal standard that you can use. However, yun nga, the disadvantage is hindi siya readily available. So still, most people will use DSS in D2O for NMR metabolomic experiments. You also can do spectral alignment. So a lot of the NMR processing softwares will have some functionalities for spectral alignment. So you can have global alignment. That means using the particular algorithm involved, uh, the whole spectra is adjusted. If you're comparing like several spectra, so all of them will be aligned in terms of like the chemical shift. In some cases, even though if you perform global alignment, there might still be like regions of the spectra that is not yet well aligned, and so you can still further do local alignment. Pero selected lang yung mga softwares that can do this, and I think one of them will be your Mestranova. So MNova NMR has this functionality for local alignment of certain peak clusters. If there are pH related shifts in Chemometrics, like one of the processing softwares that we use, having pH indicators like imidazole, formic acid, creatinine can help you adjust as well the chemical shift of some metabolites that are commonly affected by changes in pH, and therefore it will be easier for you to identify them. In some cases, or in order to also avoid these types of pH related shifts, you can actually just use a buffer solution to control the pH of your samples, and so this will help prevent these pH related shifts. Okay, so in addition to that, there are other pre-processing steps. So common pre-processing steps in NMR will be, of course, your phase correction and baseline correction. Aside from that, in cases wherein there are some protein interaction effects, you might need wherein there is some line broadening, so you might have to do some line width adjustment. You can delete certain regions that are associated more with these different like your solvent peaks, your water, so your HOD peak. So you would want to delete that as well as your urea peak in urine samples, and in order to also prevent the radiation damping. So para mas ma-highlight mo yung signals, there are also some pulse sequences that can allow you to suppress some of these peaks. But if not, if you were not able to do it, you can do region deletion before you proceed on with the rest of your data analysis. Okay, so softwares for NMR data preprocessing, of course, though there are those that are more based um on the instrument that is used. So top spin will be for Brooker. I think you can also use it for the analysis of um, the other data formats that are from other vendor or other manufacturer. Um, you also have your OpenV NMRJ. This is mainly suited or uh, mainly used for um, spectra or NMR data file that has been obtained uh, from the VNMRJ platform of Agilent or Varian NMR. So Agilent has already discontinued their NMR business. So the software that is used for data acquisition, which is VNMRJ, has been converted into an open source software. So this is your Open VNMRJ. um, and as mentioned, you have your Mest Renova suite of processing softwares, and they have specifically their own suite for NMR processing. You also have ACD Lab, so they have their Spectrus for the analysis of pre-processing and processing of your um, MS, NMR, IR, other spectroscopy data. Lastly, is Kinomics. So Kinomics is actually um, specific for NMR. It has functionalities that allows you to process as well as to profile. So later on, I will show you like how you make use of Kinomics for profiling. So it allows you to perform quantitation on the data that you acquired. And it has capabilities for you to build your own library. So you'll be able to add on um, compounds that are from your own analyses that may not be present in any databases by making use of your compound builder, and subsequently you can build your libraries using the library manager. So this one is actually a very useful tool for metabolomics. So this will allow you for targeted or quantitative profiling of your NMR spectra. And I think this is not exhaustive. There are some softwares that you can also look into in terms of like mixtures analysis. So I think Brooker has the Amix, or the analysis of mixture. The abbreviation is Amix for um and Geol will also have their own softwares. I apologize. I haven't really used the NMR yet in CIF, so they will also have its own set of softwares. Um, okay, so that is for the NMR data processing. Usually in NMR, data is either binned or profiled. So when we talk about binning, the whole spectra is divided into bins. So you can divide bins of either 0.04 or smaller 0.02 ppm, and then you get the area for each bin. So the total area within each bin will be the one that is used for the analysis. And this allows you to compensate for minor changes in line width and chemical shift. So I've mentioned earlier, certain in certain cases you can have protein interactions that can affect your line width. You can also have pH changes that can also affect your chemical shifts and so forth. And so this spectral binning can compensate for a bit of this effects. And you can also do relative concentration based on the total area. So you can obtain relative concentration information based on the total area, and internal standard is not required. So you don't perform absolute quantitation. So you can just do relative quantitation based on total peak area. And so, yun, hindi kailangan yung internal standard. On the other hand, meron tayong mas quantitative approach, which is profiling. And it makes use of signature NMR profile for each compound for identification and quantification. So so we know that in NMR, for example, your glucose, your creatinine, they will have specific signals at specific chemical shifts of specific multiplicity. So for example, you will have a singlet at 5.23 ppm for glucose. But aside from that 5.23 peak, glucose will have a whole cluster of peaks in the 2 to 3 or even 4 ppm region. So they have these characteristic peaks or peak clusters. And these can be used to profile and quantitate um, your uh, your compounds based on the NMR spectra. And usually, this quantitation is based on the internal standard. So it is known that with NMR, you can get linear response in terms of like using internal standard for the purposes of quantitation with respect to your metabolite signals or your compound signals. It can account for overlapped metabolites also. So um, it one problem also, as mentioned in our previous session, where we could talked about the advantages and disadvantages of the different instruments. NMR usually, if you have compounds that are quite similar, having similar um, signals at a particular chemical shift region, then they will be highly overlapped in that particular region. And so it is very hard to distinguish which is which if you just rely on chemical shift information. And this particular profiling using genomics, as I will show you later on, can actually account for this overlap and still allow you to do quantitation. And the data for chemometric analysis is limited only to, however, yun, yung data that will be obtained is limited to the quantified metabolites. So unlike in spectral binning, you make use of the whole spectra for your data analysis. For your chemometric analysis in profiling, it is limited to the quantified metabolites. So if you only quantify 20 metabolites, your whole chemometric analysis will be only based on those 20 metabolites. Whereas if you make use of the spectral binning, your chemometric analysis will be for the whole range of whatever components are present that has signal in the spectra. So this is just to illustrate. So this is binning. So spectral binning, you divide your NMR spectrum into small bins of about, in this case, 0.02 ppm. And so you take the area for these particular bins, and subsequently it will be converted to something like this. So you see there is a loss of resolution with increasing bin size. So usually you have to, the maximum that is used for NMR metabolomics is 0.04 ppm bin size. If Usually for my case, I prefer to use 0.02 ppm so that you don't lose a lot of the, you don't lose so much on the resolution, especially if you're using 600 megahertz NMR. You don't want to later on make use of too big a bin size that you generate data similar to 300 or 400 megahertz NMR. So you have to optimize this based on your data. And one disadvantage, as mentioned earlier, if you make use of spectral binning, if there are overlapped NMR signatures or NMR peaks, then you won't be able to tell them apart in the spectral binning. And so you make use of profiling in order to tell apart those overlap peaks. And this is just a screenshot of the use of Kinomics Profiler in order to do metabolite profiling. And from here, you will be able to see. So, for example, this is the peak clusters for your glucose. You'll be able to match it versus the actual experimental spectrum. And from there, you'll be able to get quantitative data. So you can get the concentrations in comparison to your chemical or internal standard. So in this case, DSS. So again, you get quantitative data, and para ka nagbubuo ng puzzle actually. So you can match, you try to adjust these peak clusters accordingly. Um, I won't base my quantitation on these peaks kasi nga meron siyang iba't ibang peaks that might also be overlapped with this signal. So I'll make use of the 5.23 ppm peak to adjust yung actual height of the signal for that. And that will correspond to changes also in this region. And from there, you'll be able to like, okay, see ano ba yung contribution of glucose in this particular region. So it's actually fun to do para ka nagbubuo ng puzzle. Pero if you're working on hundreds of samples, then it's a bit too manual also. So but yeah, this is it's quite fun to work with. Now, in MS-based data naman, it involves a different um, range of pre-processing. So, ang MS-based data, especially if you're looking at those data that has been generated together with a separation technique such as GC or LCMS, what you get is three-dimensional data such as this. So, you have three-dimensional data wherein you resolve peaks based on mass to charge as well as based on the retention time and the retention time. So um, data preprocessing is usually done on two-dimensional data. So the first dimension will be the MZ versus the relative abundance at a particular time. So at a particular time, such as like for example 18.5, ano ba yung MS spectra mo? You will have different values, different signals across your different MZ. And so yun yung magko-consist of your MS spectra. On the other hand, you also have the dimension of retention time versus the relative abundance for a particular MZ or for all MZ. So for example, for this particular MZ, like at 200, so meron kang um, uh, extracted ion chromatogram or meron ka ma-generate na ion chromatogram for that particular MZ. Or in the case, if you're looking at all MZ values, then that will be your total ion chromatogram. Okay, so this is just the pre-processing steps that are involved in LCMS-based data. Actually, it will also be applicable to your GCMS-based data, but there is like another step that we'll talk about for GCMS. So basically, you have your raw LCMS data. You have to perform mass recalibration. Um, it can be using an internal standard or some signal that you can assign in your software. You perform centroiding. When we say centroiding, so usually in high resolution mass spec, you get profile MS data. So that means your signals are collected continuously across. So your signals are collected continuously across through time at a particular interval, and so you will generate a profile MS data. So at very small intervals of MZ, you will get a signal. It's continuous yung scan. But this will generate a lot of data. So masyadong malaking data file yung ma-generate mo if you scan across the MZ. So for this particular 300.4 peak, if you make use of profile MS data, so ito nga, continuous yung data na makukuha mo. That's very specific peak widths of the, depende na rin sa resolution ng instrument mo and your settings. But yun, if you have too much data, the data files are very big, it's very hard to process them. And so usually we make use of centroided MS data, wherein these continuous peaks are converted to like these thin stick or line peaks. And these will be used for subsequent steps of data analysis. And then you can perform feature detection. So this is part again of the algorithms that is usually used in MS-based MS-based data. You define filtering information. If you make use of a certain threshold, so a certain signal to noise threshold can be used. If you can also define, okay, if this peak is present only in a certain percentage of my whole data set, then you can also choose what you want to do with it. Feature grouping will be more to group similar or similarly behaving ions for a particular retention time. So those that are corresponding to as the same metabolite, but just like, for example, neutral loss, or they are like adducts of are different adducts, then they will be grouped accordingly. You also have your feature annotation, which will be to provide you with like, for example, what adduct is formed, and it can also provide initial or tentative identifications based on database search, based on the accurate mass, etc. And at the end, you will have to do alignment. So your chromatograms will be aligned accordingly, either based on a particular internal standard, or based on certain algorithms that has been employed for MS data processing. Um, Again, as I mentioned, medyo mas iba yung processing for GCMS data. So, kasi, di ba, GCMS data with electron ionization, you get fragments right from the start. So, after it has been ionized, nafa-fragment na siya, and that is what goes through to your MS. And so, in the cases wherein um, in cases wherein you have overlap peaks like this, you need to be able to determine ano ba yung spectra, or yung spectrum for peak 1, ano ba yung spectrum for peak 2, ano ba yung spectrum for peak 3. And that is what is done in the convolution for GCMS. And so there are several softwares, but one of the more popular ones for this, or one of the more popular algorithms for this, will be the AMDIS, which is your automated mass spectral deconvolution and identification. So this can be downloaded from the NIST library, NIST website. Again, this is usually incorporated in the softwares also already, especially for the vendor specific software. So incorporate na rin nila to for GCMS. So for LC GCMS processing, there are of course your vendor-specific processing softwares. So may kanya-kanya silang specific processing softwares. Um, Personally, I've mainly used more of like the SciX ones, like Marker View and Peak View for mainly Marker View for data pre-processing. Agilent will also have Thermo Waters. They will have their own um, processing softwares, or it can be part of the acquisition softwares na. So depende. You also have your open source softwares. So dito na papasok yung mga those that are freely available, like XCMS or XCMS Online. So merong XCMS that is mainly based on our environment, and you also have XCMS Online, which is very user friendly, and it is web based. So as long as you have the Wi-Fi connection, you can upload your data and let XMS do the data processing online or on the cloud. Other softwares will include your OpenMS, MZMine2, and Metalign, and there's still a lot more. I'm not going through uh, the rest of the. We will not go into so much details for this part. But these are the more commonly used ones. There are still newly upcoming uh, softwares that are being launched. But yeah, so far from what I know, uh, a lot of people are using XMS Online. This is also incorporated in Analyses, so we'll talk more about that later. Okay, so with advances in analytical technologies and with the use or with the advent of the omics platforms, like we've talked about genomics, proteomics, metabolomics, these different omics generate a lot of data. They are high throughput, they simultaneously measure a lot of different compounds, and this leads to very large amounts of data. And so, what do we do with these large amounts of data? And in order to make sense of that, we can use chemometrics. So chemometrics is actually first coined by a professor in from Umeå University, which is Professor Svante Vold. Just a bit of trivia. Actually, Svante Vold is the grandson of a Nobel Prize in chemistry winner, which is Svante Arrhenius. So if you remember your general chemistry, your Arrhenius acids and bases, so I hope this gives you a bit gets you a bit nostalgic about your basic or general chemistry. So he is Professor Wold is the grandson of uh, Professor Svante Arrhenius. So pinangalan si Professor Wold kay Professor Arrhenius actually. So it makes use of mathematical and statistical approaches to design and optimize measurement procedures and experiments. It can also allow you, yung chemometrics can allow you to maximize the chemical information from analytical data. So it makes use of mathematics, statistics, uh, to make sense of those large amounts of data. So in chemometrics, you usually arrange your data in this particular manner. Although, of course, you can put your samples on the columns, your variables into rows. Pwede naman yon, but usually it is more commonly done such that your variables are on the column and your samples are on the rows, and you can have your class information. So yung class information will be the grouping of your samples. So for example, if you're looking at control versus disease, then your class information will be control or disease. And then your samples has to have unique sample names across variables. This is I think variables that are from chemical shift of NMR data, sorry, from spectroscopy data. But in terms of NMR, it can be your chemical shift data depending on the bin size. So you can make use of that. In terms of LCMS or GCMS data, this will be your MZ at a particular retention time. So remember, um, three-dimensional yung data that is obtained using MS. So each MZ can have peaks at different retention times. So you need to have um, you need to define for each variable what is the MZ value as well as the retention time. Okay, so um, you also have what is called data pre-treatment or processing. So meron tayong pre-processing, meron din tayong processing. So dito naman sa data processing, we already process the whole data set in order to make sure that they are comparable against each other. So first, we perform what is known as centering. So it converts the values of your samples, the values for each variable, for each sample to fluctuations around zero rather than around the mean. It adjusts for differences in the offsets of low and high abundance variables. You also have what is known as transformation. So transformation naman, the values themselves are converted. They are transformed into either using logarithmic transformation, or you can use power transformation, wherein they are raised to a certain power. So it is occasionally used if if you have large values relative to others that will otherwise dominate the analysis. So yun nga, logarithmic, replace x by log of x. When we talk about power transformation, you replace x by x raised to n. And you would also have row processing. So row processing naman across all, so you have different samples. Their relative measurements can be will be different. Sorry, their absolute intensities will be different, but you can uh, transform them, you can normalize them in order to make sure that their relative concentrations will be similar. And this will be looking at, for example, if you have slight changes in the mass that is used per sample, then you can also normalize per sample based on the mass or based on total composition of the sample in order to compensate for that slight differences in mass or in composition. Um, you also can use uh, ratio to landmarks, such as the use of internal standards. So you can normalize your samples to internal standards, or even, I think in some cases, you can make use of like um, uh, certain signals that are present in your LCMS data, like, that are not really related to your sample. Others will be like your quality control samples can also be used for normalization. And yun. So again, here as mentioned, yung constant total or the sum of the intensities. So total area can be used. Um, Column processing or scaling is also another pre-processing step. So this allows you to ensure that your variables are placed into the same scales. So kasi 'di ba, if you have very high intensity variables, their variation across different samples will also be high compared to like very small intensity variables wherein mas maliit yung variation niya. And so if you look at chemometric analysis, some of them generate their models based on variations or based on variance. And you can just imagine that if we don't do column processing or scaling, those high intensity variables can actually dominate the model. So you need to put all of your variables, whether they're high intensity or low intensity, on the same scale. And so, kaya yun, tinawag siyang scaling. So, some examples of column processing or scaling will be your unit variance and Pareto scaling. We'll go into a bit more details into that in a bit. So, first, normalization. So, again, it is applied to all samples and makes your samples comparable. It removes or minimizes variable dilution effects. Common techniques will include the use of internal standards, creatinine normalization for urine, but please note, creatinine normalization for urine cannot be used if you're looking at comparisons involving disease, kidney disease, for example, because of course we can expect that with kidney disease that your creatinine will change. So hindi siya pwedeng gamitin din for normalization. And another will be your integral normalization or total area normalization, wherein you sum up the total area across all variables for a particular sample and then use it for the normalization in order to get relative ratios versus that total area. There are still other normalization techniques. Some of them are mentioned here, like probabilistic quotient normalization, cyclic Lewis, quantile normalization, cubic spline, variance stabilization normalization. These are actually normalization techniques that has been previously used for genomics data that are also currently being explored for use in metabolomics data. Okay. In terms of data pre-processing and pre-treatment, so here listed are the different centering, scaling, and transformation techniques. What are the formula that you have to use, and what will they do in with regards to your data? When we talk about heteroscedasticity, okay, medyo big word. It talks about the presence of noise. So we expect that large intensity, um, large intensity signals or variables with large intensities will have more noise compared to your smaller intensity signals. So yun nga, scaling methods address these types of um, heteroscedasticity in our data. Okay, the commonly used in my experience in metabolomics, what we commonly use for scaling are unit variance or auto scaling as well as Pareto scaling. So yung first, yung centering, we obtain the mean across a particular variable and then subtract that mean from the value of for that variable for a particular sample. So yun nga, it allows you to center your values around zero rather than around the mean. Um, and then subsequently, these will be divided by the standard deviation in case of auto scaling. And in the case naman ng range scaling, it will be divided by the range. So your X max minus your minimum X value. Pareto scaling will be dividing by the square root of the standard deviation. Vast scaling will be divided by your standard deviation multiplied by your mean over the standard deviation. Level scaling will be divided by your mean. So more commonly used will be your auto scaling and Pareto scaling. Auto scaling in some cases might inflate measurement errors. So it can inflate noise. In order to avoid that, you can lessen the scaling by making use of Pareto scaling instead, which is makes use of, instead of using standard deviation, it makes use of the square root of standard deviation. So this just shows the effect of the different scaling and transformation. So you can see your original data. So you have a few large intensity ones, and there can be like some very low intensity signals. And again, if these large intensity signals dominate your model, then you might get false results. You might not be able to determine ano ba yung significant effects of your low intensity signals. And so it's very important to do proper centering or scaling and or transformation. So here, this just shows you the effects of centering as well as your unit variance scaling, Pareto scaling, range scaling, vast scaling, as well as the effects of your transformations. As you can see, with untreated, you have some signals that are of very high noise, high variation, and then with transformation, you convert them to you make them more comparable. The variations are only, or the standard deviations are only in a particular range that are common across uh, most of the signals. Power transformation, merong slight effect, pero as you can see, medyo mataas pa din yung standard deviation across certain variables. On the other hand, yung dito naman sa scalings, um, medyo makita mo yung effect ng range, UV, and vast scaling um, in terms of like highlighting those that are smaller intensities. UV scaling does the job. But again, of course, you have to consider that some of these low intensity signals might be your noise. So you need to like make sure that you also don't scale them up so much. So from the data processing, we proceed to the statistical analysis. So this will cover more of your chemometric analysis. So from previously, when we do statistical analysis, we mainly look at the univariate approach. So we determine the significance of one variable at a time. So we determine the statistics of one variable at a time, independent of others. Now, this does not fully utilize available data, especially if you're working with instruments like LCMS, GCMS, and you're doing untargeted analysis. Looking at your variables one at a time will be quite tedious if you're going to do it for all of your different signals. On the other hand, if you just selectively identify specific ions, then yun nga, hindi mo nagagamit yung hindi mo na fully utilize yung available data. And so, dito na pumapasok yung tinatawag na multivariate analysis. So this allows you to simultaneously analyze multiple variables or even full spectra. And this can help account for interdependencies of variables. Kasi there can be variables that are related, they can be correlated, or meron silang mga relationships involved in terms of like response, and this can actually help you to account for those. So, multivariate analysis approaches can be divided further into unsupervised and supervised analysis. So, in unsupervised analysis, you will have those uh, that can show ano ba yung mga inherent trends or patterns in the data. So, this is unbiased, so it doesn't incorporate any class information in its generation of the model. And some examples of this will be your K nearest neighbor analysis, principal component analysis, self-organizing maps, dissimilarity indices. On the other hand, supervised analysis naman, they make use of your class information. So, nagmo-model sila, or they generate the model based on the class or group information that you provide. And in some cases, magiging biased siya, and they can be overfitted, and therefore there is a need to always enforce or make use of some validation methods after you have done your supervised analysis to confirm yung reliability ng model mo. Some examples of supervised analysis will include your SIMCA, or soft-independent modeling of class analogy. You also have your projection to latent structures, discriminant analysis, or PLS-DA. And you can also have your orthogonal PLS-DA. So these are again non-exhaustive. These are just some of the more common ones. In terms of what I commonly use, it's mainly your principal component analysis. And in terms of supervised, I will usually use either PLS-DA or OPLS-DA. So I'll try to discuss more of those three in these next slides. Okay, so in principal component analysis, this actually allows you to reduce those multidimensional data to a few principal components. So um, what you can just imagine with LCMS data, GCMS data, you generate thousands of variables. And what principal component does is it looks at the variations in the data that is present. So then subsequently, it will group those variations into what is known as components. So the first principal component will be the one that can explain the most variation of the data. The second principal component will then be the next um, group, or it's the next component that can explain the next amount of variation in your data and so forth, until all the variation in your data has been explained. So it can provide you with patterns, trends. It can reveal the homogeneity of your data. In any inherent clusterings or trends, it can show. And here, just to illustrate, so you have your data set here. You have two different classes here, two different types of samples. Subsequently, from these sorry, from this data set, you'll be able to obtain a projection, and your observation will have a particular projection based on the principal components x sub 1 and x sub 2. Component 1 and component 2. So uh sorry, this is your axis, this is your principal component. So again, your observations will be projected into your component 1 versus component 2. And then you will be able to have what is known as your scores plot. So scores plot provides you the projection of your samples into the components. So dito, as you can see, you will be able to see whether there are any clusterings or patterns. So you can see clustering of your triangles here. Yung circles mo, medyo distributed siya across the three quadrants here. Loadings plot will be the ones to show you the influence of the variables in the variation. So, loadings plot will be able to tell you ano ba yung mga variables in your data that causes these triangles to separate out from your circles. So, here you have to look at the same directionality of pictures of your scores and loadings plot. That means kung ano yung mga variables that are in this, kung ano yung mga samples in this region, they are influenced mainly by the variables that are in this region. On the other hand naman, those that are here will be mainly influenced by the variables here. And so, kung ano yung mga triangles nandito, mataas sila in terms of like intensities or levels with the variable x1. Yun namang mga nandito, yung mga circles natin, mataas sila for this particular variable x3. Yung few samples natin dito, they are high in x2. So that is how you look at principal component analysis. Soft independent modeling of class analogy is a supervised analysis. So what happens is you create individual PCA models for each class, and based on the residuals of these models, you'll be able to determine kung saan bang class na bibilong yung sample mo. So it can be asymmetric, such as this, wherein a particular group of samples is isolated from the rest based on its heterogeneity, yung um, PCA class niya. On the other hand, you can also have your two different classes for if you have symmetric cases such as this. You also have for supervised analysis your projection to latent structures, discriminant analysis. So this discriminant analysis actually can be used to, for example, if you want to determine whether a particular sample belongs in a particular class of samples, then ginagamit tong PLS-DA, OPLS-DA, SIMCA. So this is very useful for purposes of, like, for example, quality control. You want to see if you have already established the model for your for good samples as part of your quality control. You would want to see later on if you input data from different samples or from incoming samples, whether they can fit that particular model. So usually yun ginagamit. But in terms of like metabolomics analysis, ginagamit siya for further trying to determine yung mga differentiations as well as the variables that causes these differentiations. So in PLS-DA, it makes use of, again, the additional variable or class information. And they try to, in PLS-DA, it tries to maximize the separation between your different groups based on the class information that you provide. Medyo may problema siya if you're dealing with high within class, or kung meron kang malaking amount of variations within your classes, then medyo magulo siyang, as you can see, the separation here of your two groups are not clean cut. So hindi siya directly nasa along T1, hindi siya along T2, not clean cut along T3. So, yun. So, um, there can medyo hindi clean yung separation, hindi madaling ma-identify kung ano ba yung mga variables that are contributing to the differentiation of your samples, kasi yun medyo hindi clean cut yung separation of your variables across the different latent structures. But nevertheless, this is useful. As you can see, pwede mo siyang separate. But again, we have to be careful kasi when you make use of class information, it can be prone to overfitting, and therefore, you need to ensure that it is validated subsequently. In terms of the variables important to the projection, this is actually used to identify significant variables that is important in differentiating your different groups. And so if your VIP value is greater than 1, then it is considered to be important. In terms of orthogonal projection to latent structures, discriminant analysis, this is a variation of your PLS-DA. But in this case, you make use of you make use again also of your class information, but you make use of an additional component or an orthogonal component to isolate variables not related to the differentiation of the classes. So kumbaga, sa PLS-DA, it just tries to separate your classes as much as possible, regardless of whether the variables involved are related or not to your class information. Sa orthogonal projection, medyo mas nililinis niya. So it separates out those variables that are not related to your class information, and it puts it into an orthogonal component, whereas those that are related to your class information will be put in the predictive component. And so makita mo dito, yung differentiation or yung separation of your two groups are actually along your predictive component. Yung orthogonal component mo, dun na pumapasok yung variations that are not related. So, as mentioned, you need to perform validation. So usually you have different types of validation that can be done. Commonly is performing single evaluation set, wherein 50 to 90% of the samples will constitute what we call the training set. So training set will be the set of samples wherein you base the model, and you will test this particular model with the test set. So this is like the more manual and common and basic validation that is done, single evaluation set. You also have your cross validation set. So what happens in cross validation set is um, your n number of samples are divided into g number of groups, or what we call cancellation groups. And the model is computed g times, and in each time, one cancellation group will be taken out, and this will serve as your test set, while the rest serves as your training set. So each cancellation group will be used g minus 1 times. And when n is used as the number of cancellation groups, it is known as the leave one out approach. So this is the most commonly used validation method. You also have your repeated validation method that makes use of the Monte Carlo algorithm, and is used for it is used to compute many models, and in each model your test set is different, and the variable and you make use of variable number of samples, and it enforces random selection. Medyo mas ano to, this is actually more robust, but it also involves um, more computing power. So relatively, medyo mas commonly performed pa rin yung cross-validation. So in terms of the chemometric workflow, usually yung mga softwares, yung isang software mo will cover your data pre-processing and data extraction. As we have seen, we have MZMine, XMS. I think XMS actually also already have XCMS Online already have like some statistical analysis incorporated in their web-based platform. But I usually don't make use of them. I still use it mainly for data extractions, and subsequently I still do some cleanup offline and make use of either SIMCA or Metabolomics for the chemometric analysis part. And yun, so meron kang software for processing. You can also have your software for the chemometric analysis. Or, and yun nga, as I mentioned, in some cases, you can make use of one web-based platform to perform everything. So it can be your XCMS, it can be your metabo analyst, or in vendor specific software will be your MPP for Agilent and so on. So again, these are the chemometric software tools that you can use. Metabo Analyst is a web-based platform. It's open source. You can do code-based analysis using R, Python, MATLAB. SIMCA is a commercial chemometric analysis software. Minitab, SPSS are statistical analysis softwares that also has some of these chemometric functions. We'll go a bit further into Metabo-Analyst. So again, this is actually a web-based platform that is mainly for metabolomics data analysis, although you can use it for the chemometric analysis of other types of data. So actually it is like a one-stop shop. So you can input certain formats of data, of LCMS data. It can do the initial LCMS spectral processing. And then you can do some functional analysis or meta-analysis. It has features or functionalities for enrichment, pathway, joint pathway analysis, as well as network analysis. If you have gene, metabolite data, it can do network analysis on those types of data. It can perform your statistical analysis, biomarker analysis. It can do two-factor or time series analysis, statistical meta-analysis. It can perform power analysis to determine what will be the suitable experimental designs, and it has other utilities. So, makita mo, yun nga, one-stop siya for a lot of the different things that you need to do for metabolomics data. And it's actually developed by the SIA group in McGill University, and actually it's part of the Metabolomics Initiative Consortium of or the TMIC of Canada. So these are developed by Professor Xia, Professor Wishart, and the rest of their group members. This is just to show you an example of PCA using Metabo-Analysis. So at the start, you'll be able to get this overview plot. You'll be able to see which components shows you the separation of your different samples. And then subsequently, you can look at your score plot. So here again, you will be able to see inherent trends or patterns. So in this case, your B and D are clustering together. So they're basically similar in profile. Your A and C are clustering separately. In terms of variables that are influencing their separations, you can look at the biplot. So you can look at the loadings plot, but for easier reference, you can also make use of the loadings, the biplot, wherein it shows you the scores your samples as well as the variables. So from here, you'll be able to see that A samples are actually high in variables like V22 and V7. Those that are your C samples are high in variables such as V42 and V27. Your B and D samples are high in variables such as V10, V24, V1. So again, reflecting the directionality of your scores and loadings. um, Metabo analysis will also allow you to perform other types of statistical analysis. So this is just for the statistical analysis module. Marami pa siyang ibang modules. So you have your biomarker module, your time series module, etc. etc. So there's really a lot of functionalities here. I will not have time to cover all of them, but just to show you, you can perform your basic univariate analysis. You can have correlation heat maps. Heat maps are actually very useful to show patterns in your data, and they are usually used for visualization of your data of metabolomics data. And of course, you will have your chemometric analysis functionalities like PCA, PLS-DA, sparse PLS-DA, and orthogonal PLS-DA. There are also some more advanced feature identification functionalities such as SAM, EBAM. I think some of them might be more suited uh, uh, for time series, but in any case, again, these are features that you can make use: heat maps, SOM, k-means clustering, and also you have a bit more of the more advanced algorithms such as random forest and support vector machine algorithms. And while you are making use of this, you also generate your R commands. So again, so actually this metabo analysis is R based. So whenever you make use of any functionalities, it is actually calling out a particular function in R, and so you can actually save this, and you can make use of this for uh, s- later if you want to like rerun the analysis. So after sorry, after the statistical analysis, you then perform metabolite identification. So in GCMS data, again, we have mentioned that you in with the electron ionization MS data that you generate, you can actually use it for matching with databases such as your NIST library. For proton NMR data, you can identify compounds based on chemical shift and multiplicity of peaks. Kapag medyo mas overlap na, pag mas complex yung mixtures, of course, that will be a bit difficult, but you can also make use of 2D NMR and yung functionalities nga ng Kenomics na profiling, which is yon sa mga analysis of mixture functionalities of softwares. You also, for LCMS naman, you can, if you're making use of high resolution MS, you can do putative identification based on accurate mass, and you can also use the fragmentation information that you obtain using MSMS. So ito, yung putative identification based on this can be for either your LC or your GCMS. But again, for GCMS, meron kang additional dimension of identification based on the fragmentation generated using electron ionization. And actually, I forgot to mention here, but another thing with GC data, you can make use of your retention time index. And when you're referring to NIST library, having that retention time index information can actually help provide you with more reliable identification of your compounds. So this is just to show you an example of like from your chromatogram, you can identify at a particular point your mass spectra for that particular peak. And from there, you'll be able to search the database, the NIST library, for possible identities of that peak. And so from there, you have to like of course you need to assess kung ano ba yung level of matching niya. The software will provide you the scores based on the match. But of course, you also need to sometimes check and confirm na ayos ba talaga yung match, because there are times that they might not be really matched in some cases. Pero so yun. Okay. So these are the databases that you can use for the identification of metabolites based on accurate mass. So you can input accurate mass, and you can search through these different databases for possible identity. Some of them will have some experimental MSMS data that you can also use for matching. They also have functionalities to allow you to input MSMS data, and yun nga, para magawa yung spectral matching mo for MSMS for further identification. So some of these will be your Metlin. So Metlin is developed by the same group that developed XCMS. You have your human metabolome database. So this one is more on the Canada metabolomics side. Um, you have your mass bank. So mass bank has a Europe and a Japan consortium. So you can make use of their databases as well. And you also have lipid maps, more for lipids. In terms of levels of metabolite identification, of course, highest level will be your identified compounds, wherein you analyze authentic reference standards or standards, and you identify based on a minimum of two independent and orthogonal data, such as your retention time information as well as the mass spectra. And you can also have your MSMS spectra. Level 2 will be your putatively annotated compound. So you can from metabolomics papers, you will see a lot will mention putative identification. So the putative identification is usually based on spectral data or similarity to data that is available in public databases without confirmation to reference standards. So yun. Level 3 will be putatively characterized compound classes, like, for example, lipid classes. They are unidentified, but the data available allows the metabolite to be placed in a compound class. So, for example, presence of certain functional groups will be attributed to a particular class of compounds. And so, the presence of those functional groups can actually, or signals that can be attributed to certain functional groups can allow you to assign some of these compounds to a level 3 identification. And of course, level 4, there are um, from the many data that you generate in metabolomics, there will always be bound to be some features that you will not be able to identify, and they can be differentiating, but they cannot be identified, so they are level 4 classified, or they are unknown compounds. So, in terms of these levels of metabolite identification are actually defined by the chemical analysis working group of the Metabolomics Standards Initiative. You can refer to their publication in terms of the minimum reporting requirements for metabolomics publications. So this is actually a very useful guide if you're starting out in metabolomics, because it helps you to determine ano ba yung mga kailangan mong i-consider. It can also give you information on ano ba yung mga kailangan mong i-record, ano ba yung mga main considerations, etc. So please do check out this publication. In terms of pathway analysis, there are different softwares that you can also use. These are actually more of the open source ones. This KEGG is actually a very well-known pathway database. This is Kyoto Encyclopedia of Genes and Genome. Sorry if I have made, I'm not very sure, but it's something close to that. So that is KEGG. So it provides you with the pathways for different, the whole networks of pathways for different organisms. And it's actually a very useful database. You also have this one naman is more of the commercial uh, Qiagen Ingenuity Pathway Analysis. This actually allows you to incorporate or to integrate genomics, proteomics, metabolomics data. As you can see from here, uh, it can do network analysis, it can do multi-level analysis of your gene expression, proteomics, metabolomics. Although it comes at a very steep price. I tried inquiring for one of these, and it costs thousands of dollars to have 1 year subscription. So medyo yun, it's one consideration. You have Metapsych, also another pathway database that you can make use. And of course, you have Analyses that can perform, wherein you can perform your pathway analysis. So this is just to show you a screenshot of pathway analysis with Analyst. You can input compound lists or concentration tables showing your different variables or different compounds. And then you have your samples as well as your class information. And this can be used for for the pathway analysis. You can also make use of data that are available in if you want to look at the pathway analysis of data that's available in their metabolomics workbench database, you can also do that. um, for this one, I just inputted the example data. So if you want to practice using Metabolomics, they have some example data that you can use for practices. And so from there, you'll be able to determine ano ba yung mga significantly perturbed pathways based on the significance values, as well as the number of metabolites that are in your list, that are in these particular metabolic pathways. And from here, you will be able to have the visualization. So higher, closer to the upper right, the more important and the higher impact they are. And if you click on one of them, you'll be able to see yung diagram of that particular metabolic pathway. The information, yung number, those that are in red are actually the ones that are in your compound list. So ito yung mga nasa compound list mode that are present in that particular pathway. And the numbers here are actually the KEGG metabolite IDs. um, Recently there are a lot of there are a lot of improvements or advancements in terms of molecular networking. So LCMS, GCMS, untargeted metabolomics, you generate a lot of data, you do chemometrics, you only identify the compounds, you only try to identify the metabolites that are found to be significantly different or significantly differentiating. And so with that, you limit yourself. I mean, um, the rest of the data is not used. So kumbaga na di-discard na or nawawalan na ng gamit yung the rest of the data. But actually, especially in the era or in the area of natural products research, they can actually be very useful. So currently, these organizations try to get as much of these LCMS and LCMSMS data, and from there try to establish. Kasi di ba, when we look at fragments, these can actually have information on characteristic functional groups, and so this information, if can be useful in the identifying certain moieties present in a particular compound. Um, so in molecular networking, they make use of those data to identify, for example, in this case, for this group of stenotricine compounds, all those ions that are close to these stenotricine compounds actually have fragmentation patterns that are close or similar to these identified compounds. And so there is a possibility that they can be analogs or derivatives of these compounds, even though they are not yet identified. So dito papasok yung the use of molecular networking, and can actually be very useful in the long term, because from here you will be able to build databases of MSMS data, and these might lead to further identifications of different compounds by through the use of molecular networks. Um, okay, so the last part of the metabolomics workflow is of course validation. It is good to all to have biological experimental validation of your studies. It helps to corroborate preliminary results and consolidate the biological interpretation of your studies. You can have external validation wherein you make use of an entirely new set of samples that are collected and processed, or you can also have internal validation wherein samples that are obtained from the same study can be subjected to targeted analysis. So kung initially nag-conduct ka ng untargeted metabolomic study, you have identified the significantly perturbed metabolites and subsequently the pathways. You can then do a more focused analysis or a targeted analysis of those specific pathways and metabolites using the same set of samples. So that is internal validation. You can also have independent specific studies. So for example, if you have done an in vitro study, those metabolic pathways that you have identified to be significantly differentiating, you can look at it in an in vivo or in humans, and vice versa. So these are the different types of validation techniques that you can make use of. And so please do stay put. We have one more speaker coming up. So we'll be hearing from Dr. Rod Sumayaw on his applications of metabolomics in clinical studies. So that will be a very exciting presentation up ahead. Acknowledgements, of course, much thanks again goes to the OST, PCHRD, and the Balik Scientist program for sponsoring my Balik Scientist journey here and allowing me to be part of this program. It is an honor. It's a very it is a privilege, and I'm really grateful for that. De La Salle University Manila for hosting me, for Professor Drexel Camacho for being the one who has invited me over and has introduced me to this Balik Scientist program, the LSU CIF staff who has been behind the scenes on all of the works involved in this webinar series. Thank you very much. You guys are really great. And shout out to Chris, to Alona, to Hazel, Jeka, Jude, Sir Veer, Sir Jojo, to the whole team for all the work that you do for this webinar series. To our guest speakers, Dr. Pekka Lingasyon, Dr. Melo Syria, and Dr. Rod Sumayaw for agreeing to be part of this webinar series. To the research control, I just would like to give a shout out. Uh, I'm not sure whether this will reach them, but a big shout out to researchers and consortiums working towards open source databases and software solutions for metabolomics. A lot of researchers will not be able to do their work given, like, of course, funding issues. So these open source softwares and open source databases are very useful and very instrumental in a lot of the work that we do. And of course, much thanks to families and friends for the ever constant inspiration and support. And lastly, thanks be to God. All praise and thanks be to God for His wonders and His grace. And that's all, folks. If you have any questions, if you need any help, if you want to pursue metabolomic studies, and you need help, please do let me know, and I'll be very much happy to help out. So that's all.